ASH 2018 has really been an AML meeting, which is super fun for an AML investigator because we've had many years with not that much to talk about, and then the last few have been a significant improvement, culminating in several recent approvals this year. My job today is to talk about IDH at this meeting. So certainly the IDH 1 and 2 mutations have been topics of discussion in many different sessions. As the audience may know, this brings back flash memories of the Krebs cycle, which kind of makes everybody stressed. But basically, IDH 1 and 2 mutations confer neomorphic enzymatic activity, which results in accumulation of the oncometabolite 2HG. 2HG then results in epigenetic changes and impaired cellular differentiation. And as many people know, this pathway has been extremely exciting because IDH 1 and 2 mutations are present in AML, and there are inhibitors of IDH 1 and 2 that are commercially available in the United States for patients with relapsed disease. Ivosidenib for patients with IDH 1 mutations and enasidenib for those with IDH 2 mutations. I presented today on an interesting group of patients with IDH 1 mutations and untreated AML. So this was a subgroup of a phase 1 study in which we talked about 34 patients with untreated AML, so newly diagnosed older patients who were treated with single agent ivosidenib. And these are actually interesting data because the CR/CRh rate was 42%, the overall response rate was 58%. And these again are patients who are treated with a single agent, oral, quite well tolerated drug and going into complete remission. So this has led to a lot of discussion. First of all, how to prospectively identify those patients who might respond to single agent therapy. And secondly, in light of the recent approval of venetoclax in combination with hypomethylating agents, how do we distinguish those patients with newly diagnosed disease and IDH mutations who should be treated with single agent inhibitors, should they become approved and available, versus venetoclax combinations? And I think there are a variety of considerations that will go into making this decision, but it's certainly an exciting time for AML and a particularly interesting one for IDH 1 and 2 mutations.